This video is produced by Sammy and
Kevin Burgess
for byinquisition Press to support the workbook Sophomore Organic Chemistry 2 by Inquisition, available from www.byinquisition.org.
Hydrolysis of amides is critical in biological chemistry because of enzymes called proteases. Proteases are everywhere inside the cell in viruses, bacteria. They cleave amide bonds in peptides and proteins. Without understanding the mechanism of hydrolysis of an amide under chemical conditions, we can't hope to understand what proteases do. When we protonate an ester, where does it protonate?
The carbonyl oxygen.
And why does it do that?
Because then resonance from the alkoxide would be possible.
Similarly, when I take an amide, where will that protonate? Will it protonate on the nitrogen or the oxygen?
Definitely the oxygen.
Because if it protonated on the nitrogen, there wouldn't be a resonance structure. That's important when we look at the hydrolysis of amides. The first step under acidic conditions always going to be protonation of the carbonyl oxygen. Attack of water, loss of a proton, gain of a proton, and finally loss of ammonia for a primary amide, loss of ammonia, and loss of a proton will give the product. For benzamide, we can represent the mechanism again as protonation, attack of water, proton transfer, loss of ammonia, and finally generation of the product. If I take the primary amide of lactic acid and hydrolyze it, I get back to
Lactic acid.
And if I take an amide from valine, I would get back to
Valine.
valine itself. Proteases allow us to take a chain composed of sometimes hundreds of amide bonds and cleave selectively at one place. One half will be
Carboxylic acid.
and the other half will be an
Amine.
amine. Let's go back to the basics and look at how an enzyme might achieve this. To get from an amide to an amine plus an acid, we jump over a barrier, and that barrier is called a
Transition state.
good, and then we fall down into a well, which is called a
Intermediate.
and jump over another barrier,
Another transition state.
and finally to the product. If an enzyme wants to do its job, there are actually two ways. One way is to lower the highest energy barrier in the process. What does that do to the rate of the reaction?
It increases the rate of the reaction.
Yes. Another way to reduce the size of the energy barrier would be to destabilize the amide. In fact, what is the most common way for enzymes to increase the rate of hydrolysis?
By decreasing the activation energy.
Yes, by bringing down the energy of the transition states. Let's say we have a protease which is new and unknown to us, and we would like to know, does it cleave at the C-terminal amide of a peptide sequence? A clever way to do that is to attach a precursor to a fluorescent dye to a library of peptide and select the one in the library that is actually cleaved by the protease. Only that peptide will give you the fluorescent label. Nitrile hydrolysis is related to amide hydrolysis. Protonate on the nitrile N, attack of water, loss of a proton, tautomerization gives a product. So if I color in a water atom and ask where did the atoms of water go, they would go one on the carbonyl and two hydrogens on the nitrogen. We can do this reaction backwards. And to do the reaction backwards, we need a good dehydrating agent. Simple math tells you if you take phosphorus pentoxide and the right stoichiometry of water, we generate phosphoric acid. Sammy, can we take a secondary amide and generate a nitrile from that?
No, because we can remove an oxygen and then one of the hydrogens, but there's no other hydrogen to remove water.
Working backwards, you could imagine what amides might be used to generate benzene nitrile, or propionyl nitrile, or even more complicated nitriles with other functional groups in them.